Hey everyone, we all know that organs are complex structure formed of numerous tissues, where one group of tissue produce the signal that induce the another group of tissue to change its behavior. The tissue which induce the change in other tissue are known as the inducer, and the tissue being induced is known as the responder. For responding to a signal, the responder should be competent. In today's video, we are going to discuss about induction, types of induction, competence, and what are the factors that make the tissue to be competent with examples. Hey everyone, welcome back to BIOSKUL. I'm Gayatri and today's topic is induction and competence. At the end, we are going to solve some previous year CSIR NET questions related to this topic. So here we begin. What is induction? We know that the induction is a specialized set of instruction to a group of cells from another group of cells. And there are at least two components to every inductive interaction. What are those components? The first component is the inducer. Inducer means it is the tissue that produces the signal and that changes the cellular behavior of the other tissue. Okay, so it induces changes in cellular behavior of other tissue is known as the inducer. And the second component is the responder. What is responder? Responder is the tissue that is being induced. Okay, responder are the tissue that is being induced. For responding to a particular instruction, the responder, it must be competent. So now, what is competence? Competence is the ability to respond to a specific inductive signal. So how the cells have become competent? They become competent by expression of appropriate receptor. Okay, and the best example of induction and competence is the eye lens development in amphibians. So let's see the example. So here in the diagram, if the optic vesicle is placed in ectopic location in the head ectoderm, then it will induce the head ectoderm to form the tissue. Okay, so here if we place the optic vesicle in the head ectodermal region, then what will happen? The optic vesicle, it will form the lens, but not all the tissue respond to the signal produced by the inducer. Here, if that optic vesicle is placed in the trunk ectodermal region, then it cannot form the lens. Why? Because the trunk ectodermal region is not competent. Okay. And third one, if optic vesicle is removed, no lens is induced because the optic vesicle is the inducer which produces the signal. If optic vesicle is removed, then how can lens be induced? Okay. Tissue other than optic vesicle is implanted, then no induction occur. So now the question is, how the head ectoderm is competent, not the trunk ectoderm? The question, the answer is that the head ectoderm or the inducer synthesize the OTX2 transcription factor in the head ectoderm. Okay. So it induces the expression of OTX2 in the head ectoderm. As the neural folds rise, inducer from the anterior neural plate induce expression of PAX6 in the anterior ectoderm that can form the lens tissue. Okay, good. First, what will happen? As the inducer, it will synthesize the OTX2 transcription factor in the head ectoderm. Then PAX6 from neural plate. Okay. Then then the neural plate induce PAX6 expression in the anterior ectoderm that can form the lens tissue. So, expression of PAX6 protein constitute the competence of surface ectoderm to respond to the optic vesicle. Okay. So, why the head ectoderm is competent and respond to the inductive signal from optic vesicle? Because it expresses the protein, and that protein is known as the PAX6 protein. Okay. Then the optic vesicle, it secretes the BMP and FGF family paracrine factors. Here you can see this optic vesicle, it synthesizes the BMP and FGF family paracrine factor that synthesize the SOX transcription factors, and that help in the induction or the formation of lens. So now we know that the PAX protein, it is important in making head ectoderm competent. Okay, so let's see some examples. In the optic vesicle, the PAX6 is wild type as well as in the surface ectoderm. So what do you think? There will be the formation of lens or not? Yes, there will be the formation of lens, or the lens induction will take place. And the second option, the PAX6, it is mutant in optic vesicle, but wild type is present in the surface ectoderm. If the PAX6 is wild type in the surface ectoderm, then the surface ectoderm is competent and it will develop the lens. And option 3, PAX6, it is wild type in optic vesicle. There is no need, because PAX6 should be wild type in the surface ectoderm, so that the surface ectoderm will be competent and it will help in, it will develop the lens. But here the PAX6 is mutant in surface ectoderm, so it cannot develop lens. Similarly, in option 4, the PAX6 is mutant both in the optic vesicle and the surface ectoderm. The one point you have to remember that for the lens induction, PAX6 should be wild type in surface ectoderm. Whether it may be the wild type or it's the mutant type in optic vesicle, that does not matter. The thing that matters to form the lens is that the PAX6 protein should be wild type in surface ectoderm. But here it is mutant, so there will be no lens induction. Okay. Now comes to the types of induction. There are 4 types of induction. Okay. So what are those? The first one is the instructive or permissive induction, and the second one is sequential induction, third one is the reciprocal induction, and fourth one is the regional specificity of induction. Now we are going to discuss each type of induction. So the first is the instructive or permissive induction. So what is instructive induction? When one group of cell instruct the other group of cell to acquire a particular trait or fate is known as the instructive induction. Okay. The best example is the lens development. When the optic vesicle is placed under a new region of the head ectoderm, that cause that region of ectoderm to form the lens. Here the optic vesicle, it instruct the ectoderm, head ectoderm, to form the lens. That is the instructive interaction. Okay. Then what is the permissive? Permissive means to give permission. So when the responder permit the inducer to induce any change is the permissive induction. Okay, to give the permission to induce any change is known as the permissive induction. Okay. Then now the second type of induction is the sequential induction. What do you mean by sequence? Sequence means it's a series, right? So the sequential induction involves the production and release of series of chemical signals. Okay, and that multiple chemical signals are required to form a particular pattern. So let's have an example on sequential induction. We know that the optic vesicle, it induces the ectoderm to form the lens, and that lens can also induce the ectoderm above it to form the cornea. Okay, so lens, it will induce the ectoderm to form the cornea. So the signals from the lens induce that corneal ectoderm to become the columnar ectoderm, and it secretes the collagen. Okay, so in the first step, what happened? The lens, it induced the ectoderm above it to become the cornea, and the signal from the lens, it induced the corneal ectoderm to become the columnar ectoderm, and it secretes the collagen. And in the next step, neural crest use the collagen matrix, and it secrete a set of protein like hyaluronidase, which help in the further differentiation of cornea. Okay. Then after the differentiation of cornea, a third signal that will synthesize the hormone, that hormone is known as the thyroxine, which help in making the cornea transparent. Okay, and it lead to the complete development of cornea. So for the complete development of cornea, the multiple signals, they act step by step, and this type of induction is known as the sequential induction. Okay, so now comes to the third type of induction. The third type of induction is known as the reciprocal induction. Reciprocal means like a cell which is act as an inducer at a point, and at the second point the cell, it becomes induced. Okay, so that is the reciprocal induction. When both the type of cell act as a both inducer and a responder, then this type of induction is known as the reciprocal induction. So the first step, what happens? Optic vesicle, we know that optic vesicle, what will it does? In the optic vesicle, it will induce the head ectoderm to form the lens. So after the formation of lens, then the lens, it will induce the optic vesicle. In the first step, optic vesicle is the inducer, and now the optic vesicle is induced. That is reciprocal induction. When the lens induces the optic vesicle, it forms the optic curve, and then it differentiated into two type of retina, which is known as the pigmented retina and neural retina. And the fourth type of induction is regional specificity of interaction, or it is also known as epithelial mesenchymal interaction. As its name indicates, epithelial mesenchymal interaction means it involves the interaction of sheets of epithelial cells with its adjacent mesenchymal cells. It involves the interaction of epithelial cells with the mesenchymal cells. And the important point is that the mesodermal cells here instruct the overlying ectodermal cells to acquire the fate of epidermis. So what will be the fate of epidermis? That will be determined by the mesenchymal cell. Okay. So here in the diagram, okay, these are the source of mesenchyma, and here is the epidermal epithelium. So what will be the final fate of epidermis? That will be, what will be the final fate of epidermis? That will be determined by the mesenchymal cell. So here you can see here the source of mesenchyma is wing. So it induces the formation of wing feather. If the source of mesenchyma is thigh, then it induces the formation of thigh feather. When the source of mesenchyma is foot, then it will help in the formation of scales and the claws. So in regional specificity, the final organ depends on the origin of mesenchyma. Okay, this is very important. The final head depends on the origin of mesenchyma, not the ectodermal cell. Okay. Now comes to the previous year CSIR NET question related to this topic. What is the first question? The first question is instructive and permissive interaction. We have discussed about the instructive and permissive interaction are two major modes of inductive interaction during development. The following compares some properties of cell lines and cord blood stem cells. So let's see what are the properties of cell lines and cord blood stem cells. The cell lines, which are stored in liquid nitrogen, can be retrieved for experiments where they behave as per their original self. Okay, so the point is that the cell lines, they behave as per their original self. The cord blood can also be retrieved from liquid nitrogen for procuring stem cells. Unlike cell lines, stem cells can be additionally induced to undergo differentiation into desired lineage, which are very different from their original cell. So here the cell line, it expresses the original self, whereas the stem cell is different from the original self. So what is the instructive interaction or instructive induction? So in the instructive interaction, a signal induces a group of cells to express the new set of the genes. So the stem cell, it shows the instructive interaction. Okay, the stem cell, it will be so instructive interaction, whereas the cell lines, they produce what they have. Okay, like they are by the permissive induction, or in the permissive interaction, what happens, they express their original fate, but for that they require the environment which is provided by a signal or the instructive signal. So here the cell line, it shows the permissive interaction. Okay, so where it is, yeah, option 3 is the correct answer. Cell lines show the permissive interaction, whereas stem cell, it shows the instructive interaction. Question number 2. In chick development, wing feather, thigh feather and claw depends on epithelial specificity conferred by induction from mesenchymal components from different source of the dermis. This may attribute to is the direct question, and we know the answer. The answer is the regional specificity of induction. Option B is the correct answer. Question number 3. Lens formation requires sequential events whereby anterior neural plate signals the anterior ectoderm to promote secretion of PAC6, which render the anterior ectoderm more receptive to secretion from the optic vesicle. The above can be best explained by which of the following phenomenon? The lens formation. The best is induction and competence. During lens formation in Xenopus, following statement have been proposed. Okay, let's see what are the options. Lens induction can be achieved in absence of optic vesicle. Never. So this one will be the wrong. Optic vesicle can induce presumptive trunk ectoderm to form the lens? No. Optic vesicle, it induces the head ectoderm to form the lens, not the trunk ectoderm. So this one will also the wrong answer. Only the head ectoderm can respond to direct signal from optic vesicle from the lens. Yes, is the right answer. The anterior neural plate primes the head ectoderm via BMP4 and FGF8 prior to signal from the optic vesicle. Yes, it's right, and we have discussed it more details in our slides. Okay, so the option C and option D are the correct answer. Which of the above combination is correct? Is one, that is C and D. The next question is, the functionality of PAX6 gene in the formation of optic and nasal structure may be attributed to the following. Okay, PAX6 makes the optic vesicle competent and allows the lens formation. No, PAX6 makes the head ectoderm to competent, not the optic vesicle. So this one is wrong. Optic vesicle can induce any part of head ectoderm to form nasal and optic structure. Due to the presence of PAX6, optic vesicle, it induces only the lens formation. So this one will be wrong. PAX6 render head ectoderm competent to receive signal from optic vesicle. Yes, it's true. Apart from the optic vesicle, head ectoderm may also be induced by BMP, FGF. So PAX6 is not exclusive for lens formation. I know that the OTX2, it will be expressed by the head ectoderm, then the neural plate, it will synthesize the PAX6. PAX6 is required for competence of head ectoderm to induce the lens formation. By optic vesicle, it will synthesize the BMP and FGF family members that will help in the expression of SOX transcription factor, which will help in lens formation. So the option D is the correct answer. Which of the above attributes are true? The C and D, that is option 2 is correct answer. Thank you for watching my video. If you find this video very useful and you are able to solve questions related to induction and competence, then please like, share, and if you are new to my channel, then please don't forget to subscribe my channel. Bye bye. See you in my next video.